Hello, hi, a very happy good evening. So this is Krishnaveni and we've been talking about the story of life on earth, and that's biology. Yes, welcome to my class. Hello Vishal, hi, a very happy good evening. So thank you for joining my lecture. So please do like, share and subscribe. So welcome to everyone who's here watching my session. So today we are in lecture 3 talking about inheritance of 2 genes. So long I was talking about inheritance of 1 gene and I have solved enough problems. So today we move on to inheritance of 2 genes. So today conceptually we are going to talk very less, but we are going to solve a lot of problems, because problems is where you people go wrong, right? So I have almost set of 15-20 problems that we will solve. Okay, yes Vishal, a very happy good evening. All right, so I hope all of you had a very nice weekend. Yes, everyone is fine and well. So shall we go into today's lecture? So before we go into today's lecture, so we are meeting once again this evening at 6 p.m. So we are talking, or we are winding up the chapter, or we are discussing important questions from the chapter Cell, the Unit of Life. So please join me for the same. Okay, so we are talking about Mendelian genetics, inheritance of 2 genes. So please pay a lot of attention, because the questions are where you guys make wrong or where you guys lose confidence. So we'll make sure we will solve all the questions in detail, and I'll make sure you have all understood this properly. All right, yes. Okay, so before we go into today's lecture, so the announcement that I want to give you. So today we are talking about, or we are discussing important questions from the chapter Cell, the Unit of Life. So we are seeing this chapter, we are discussing important questions at 6 p.m. this evening, that is after a break of 1 hour. So please make sure you join me, because this can be tricky as well. So Cell, the Unit of Life, though it has less weightage, so it can be tricky, so don't leave marks unnecessarily. So that is my point. So please join me for the same for this chapter. All right, yes. Okay, so with this I move into today's lecture. So please join me at 6 p.m. So please do like, share and subscribe the channel. So share the link with your friends so that they also benefit, because your NEET exam is nearing. So every single concept, so every single thing that you read today, so it might be useful. So might be now if I teach you and if you listen, might be your concepts are clear. So there's a lot of things like that. So please make sure you join me. All right, yes. Okay, so with this I want to say one more thing. So today is World Blood Donor Day. So that is 14th June is celebrated as World Blood Donor Day. So how many of you here are above 18? Has anyone tried donating blood? Yes. So now more than blood donation, it is plasma donation that is very famous because of the pandemic. Amidst the pandemic, people are looking for plasma. So what is plasma and what is blood? Yes, so we are biologists, right? So we should know what is plasma and what is blood. Yes, so plasma is the liquid portion of the blood which has no WBC, no RBC, no platelets. But blood is the complete thing. It has both the plasma, the liquid portion, as well as the cells. Alright, so that is the difference between plasma and blood. So when I say World Blood Donor Day, so it can be, you can either donate the entire blood, you donate it in units, okay. So unit of blood is known as pint, P-I-N-T, okay. So you can donate the entire blood including plasma and your all the components of your cells. Otherwise you can donate only plasma. You can donate only RBC, you can donate only platelets, because people are also in need of platelet. So they also donate only platelets, alright. So it can be done in either way. So that is World Blood Donor Day, and the host for this year is Italy. So please donate blood and keep the world beating is the theme of this World Blood Donor Day. Okay, so some of the facts that I found, okay, so it might be interesting for you. So just one donation can save up to 3 lives. So every single donation that you do, you can save up to 3 lives. So your tears cannot save the world, but your blood can save the world. So that is a beautiful quote that I saw on World Blood Donor Day. So one single donation can save up to 3 lives. So it is said that the more you donate blood, the more perfect or more healthy life you lead to. Okay, I'm really sorry, your chat is not visible. So there's a technical issue and they're getting through. So once it is ready, and I will be able to see your charts. Okay? Yes. So just one donation can save up to 3 lives. So more than 38,000 blood donations are needed every day. So we are all in need of a lot of blood donation today. So if every citizen who is responsible enough to take care of his blood, okay, goes forward to donate blood, then that is worth many things. Okay? Right, so this is one thing that I want to tell you. Yes, hello, hi Vishal, hi Titan, hi Shimpi. Yes, a very, very happy good evening. Hi Keshav, hi Satyendar. Yes, perfect. So you guys were answering for my question. Okay, so only now I'm able to see the chat box. I'm sorry. Okay, but now is my voice and video are clear? Yes, Zarani, very, very happy good evening. So hi Antonia B. Okay, so the video is lagging. Now I guess everything is fine. Yes, okay, fine. So you can donate whole blood every 56 days. So if you are donating the entire blood, including your cells, your plasma, everything, okay, so you can donate blood every 56 days. You can become a member of your nearby blood bank of your city or your town. So that makes a lot of difference. You're doing something good. So when you do something good, you yourself feel a lot better, a lot confident. So you can donate blood every 56 days, and we encourage you to donate as often as possible because you will stay healthy. Fine. So donating whole blood, that is all the components of blood, directly donating blood, takes only 10 to 15 minutes. So it is not a tedious task. So it can be done very quickly. So please try donating, or if you know anyone who's healthy enough, please encourage them to donate. Okay? Yes. So the famous donor, blood donor, is your O negative and the recipient is your AB negative. Okay. So with this, how do they collect? They collect it in anti-clotting factor. So they collect it in a bag. Okay. So that has anti-clotting factors so that the blood does not clot. So if your blood clots, then it does not make any difference, right? So they have to store blood. So that is how they do. But one more thing. If you donate RBC, so RBC has a very less lifespan. They cannot store it for a very long time. So that is also one of the issue. Okay? Yes? Okay, so here we go to lecture 3 of principles of inheritance and variation. So today I have only 2 sides for concept and the remaining is all questions. So make it very thorough because inheritance of 2 genes is where everyone goes wrong. Fine? How much ml from a person? So if you are healthy enough, they collect 5 ml blood or 1 unit. So they say 1 pint. So you would have seen that a bottle which collects blood. So your blood, how much you give, how much you donate, it is measured in pints, P-I-N-T-S. So they collect 1 pint from a particular person. Alright Keshav? Yes. So it has a particular measurement in a lot of standard measurements. You can check it on Google. So they say 1 pint of blood. Okay? Yes? Alright. So with this introduction, I move forward. So what are the topics that we were discussing? Mendel's experiment, inheritance of 1 gene, Mendel's laws 1 and 2, incomplete dominance, and we have codominance. So today we'll move to inheritance of 2 genes and law of independent assortment. For all those who are here without watching the second lecture, so please go back and watch, because in lecture 2 I have solved a lot of questions from your inheritance of 1 gene. Some questions are tricky, some questions may be simple, but yet you make a lot of mistakes. So please go through that. Okay? Yes, so now moving here, so can you quickly tell me the ratio of all the crosses that I ask you? Yes, so if there is a question which says, what is Mendel's phenotypic ratio? So what is the ratio that you will give me? Yes, so quickly tell me the ratio. So what is monohybrid cross? Genotypic ratio. phenotypic ratio of Mendel's Mendel's monohybrid cross. Yes, then I want the test cross ratio. Yes, come on quick. 1:2:1 is the genotypic of monohybrid and 3, okay, right, absolutely right. So this is 1:2:1, this is 3:1. So genotypic talks about the genetic makeup and phenotypic talks about the physical characteristic. So physical observation, so I have 3 tall plants and 1 dwarf plant. So this is capital T, capital T. So this is homozygous dominant, homozygous recessive, and this is heterozygous. Yes, what is test cross ratio? Yes, Antonia B, absolutely right. So 1:1. So test cross ratio will be this, right? So this will be the test cross ratio. Yes, absolutely right. 50% each. Very good. Then what is the ratio of your incomplete dominance? What is the ratio of incomplete dominance? Genotypic ratio, phenotypic ratio. So what is the example of codominance? What is the example of incomplete dominance? Incomplete ratio, incomplete dominance. Yes, come on quick. Same as monohybrid, so that is 1:2:1. So here your monotypic, your genotypic as well as phenotypic ratio is the same. So you have red flower, you have pink flower, you have 2 pink flower, 1 red flower and 1 white flower, right? So that is why 1:2:1, right? Yes, you're absolutely right, Keshav. Okay, so fine. So this is, next one is codominance. So what is the example of incomplete dominance? What is the example of codominance? Yes, for codominance it is your ABO blood grouping. Yes, so in your ABO blood grouping, the gene this controls is gene I, okay, and you have 3 alleles. So what are the alleles? B, A, B and I. So these 2 are dominant compared to your small I, right? Yes, Munna, hello, hi, a very, very happy good evening. Yes, I'm good, thank you. How are you? Thank you, Munna. So please do like, share and subscribe. Phenotypic is 3:1 for what? For your incomplete dominance. For your incomplete dominance, it's 1:2:1. Both your phenotypic and your genotypic ratio are the same, right? Okay, so ABO blood grouping. So these are the 3 alleles. Yes, Keshav, you're right. So here the example is mirabilis. You have your dog flower. So in your pea plant, a gene which controls starch size as well as shape, right? So that is also incomplete dominance. So this is in case of codominance. So IA for A blood group, IB here and small I, right? So IA produces A blood group, IB B blood group, II produces O blood group. So if you have IAI, so this is also A blood group, okay? Then IBI, so this is B blood group. So if it's IAIB, then it is AB. So this is also known as multiple allelism because you have more than one allele, right? So the ratios are clear. No one has any doubt? Yes? Yes, Vishal, you're right. Okay. Anti-Hirinam, okay. Sunday ko bhi class rakho. Oh my god, so one day we all get rest. So if you do not get breaks or if you do not take breaks, we will definitely break down. So it's okay. So one day you can rest and take care of yourself. Maybe Sunday we can have a fun session, but yes. So let's plan it much later because people are very seriously preparing for NEET exam. Okay. Yes, Munna? Alright. So moving forward. So what are the laws of Mendel's? So Mendel's laws are called as law of inheritance. Yes, give me the first 2 laws. Then I move to inheritance of 2 genes. What are the laws of Mendel? Yes, give me the first law. Yes, quick. Come on. So is there anyone who still haven't understood the ratios that I wrote on the previous slide? If you haven't understood, ask me again. I am ready to explain. Yes, the first law is law of dominance. Okay, the second law is law of segregation. Yes, you are absolutely right, Kesha. So law of dominance talks about your F1 generation. So only the dominant character is expressed, right? So law of segregation. So these two segregate independent of each other in the F2 generation. So these two laws are made based on your monohybrid cross, right? Yes, so I haven't taught you law of independent assortment. So that comes in your inheritance of two genes, right? Okay, absolutely right. Everyone is right. So moving forward to inheritance of two genes. Why is this a tough one? So I guess before this, all of you also know what is a back cross. Yes? So you take the F1 progeny and you cross it with either the dominant parent or the recessive parent. So if you cross it with the recessive parent, it is known as your test cross, and if you cross it with the dominant parent, it is known as outcross. Clear? Yes? Fine. Inheritance of two genes. So long in your monohybrid cross, so what are the things we spoke about? Only one character we spoke about. What is that one character? Height. Either tallness or dwarfness. Now I am going to talk about two characters of your pea plant, and that is why it is inheritance of two genes, which is also known as your dihybrid cross. So di is two, right? So here I am going to talk about two characters getting passed on simultaneously. So how is it? So what are the two characters that I chose? So this is not I, so it is Mendel who chose. So it is seed, color and seed shape. So Mendel chose these two type of characters. Wild type and mutant type, please explain it at the end of class. Alright, and Niabi, remain me at the end after I finish inheritance of two gene. Okay. So here I am choosing two characters, seed color and seed shape. So one seed color is yellow and the shape is round. The other one is green. So these are the traits. So the character that I am choosing, so by now I guess all of you know the difference between character and a trait. Yes. So character is your seed color plus your seed shape. So I am talking about two characters. Okay. So the trait is yellow with round shape. The other one is green with wrinkled shape. So how do I assign the alleles here? So yellow, round. So this is dominant for me. Okay. And this is round and green and wrinkled. So this is dominant, this is recessive. So I have designated or labeled my alleles here. Okay. So hybrid crosses are between plants differing in true traits. So now how do I write it? Okay. So what were this? So this is dominant, this is recessive. So these are the parents. So what is this? Yellow color seed which is round in shape, and this is green color seed which is wrinkled. So according to Mendel, so this was dominant, this is recessive. This is recessive, this is dominant. Okay. So how do you find the gametes here? So if it was one character, it was easy for you to find the gametes. Now for two characters, how do you find the gametes? Once again, it is very simple. So divide it like this. So for this you find the gamete, for this you find the gamete. Because this is one character, this is one character. For this character, one gamete will come. For this character, one gamete will come. So now for Y and Y, since it is homozygous condition, so one Y will come. For R and R, one R will come, that is capital R. So here one Y, one R. So these are your gametes. So when I pollinate them, so it will be capital Y, capital R, small r. So once again the dominant character is present. So it will be yellow and round. So this will be the phenotype of my F1 generation. Clear? So in my F1 generation, I do selfing. Okay? Number of heterozygous is number of gametes. Yes, you can say it that way also. So now I do selfing, meaning I cross it with the same type of plant. So here, how do I find the number of gametes? So once again you divide, but this is not homozygous, this is not homozygous. So in your monohybrid, if it was like this, how did you write? You wrote this T separately, this T separately. So here also Y, small y. So y into capital R, small y into capital R, then capital Y into small y into small r. So you have 4 gametes here, capital Y into capital R, capital Y into small r, small y into r, small y into small r. So you have 4 type of gametes from one parent. Since both the parents are identical, you have the same type of gametes here also. So how many of you didn't understand this step? Yes, let me know. So it's capital Y into capital R, small y into capital R, capital Y into small r, small y into small r. So we have these are the gametes. So how many of you didn't understand how to find the gametes? Ask me now, because a lot of problems are based on this step. Yes, ask me right now. Quick. Everyone understood? So these are the gametes of your F2 generation. Now how do I do the crossing? So I write the gametes like this. Yes, I am writing the gametes like this. Okay. Ma'am, for this type AABB, how many type of gametes are possible? Okay, wait, I will write down the gametes, then I will tell you. Okay, so this is gamete from one parent, this is gamete from another parent. Okay, so now Keshav has a doubt as to how to find the gamete of AA, BB. So both are small c, capital C. So you have 3 characters. So divide it like this. So these 2 are homozygous, 1B. These 2 are homozygous, 1C. So here you have 2 possibilities, A or capital A. So here you have 2 type of gametes. Okay, Keshav? Yes, so you have 2 type of gametes here. Is this clear? Okay? Fine. 8 gametes? No. No, you will have 8 gametes when it is like this. You will have 8 gametes when it's like this. So here you are having 4 gametes because it is 2 power 2. Here it is 2 power 3 when you have 3 homozygous characters. If it is heterozygous characters, this is the formula. If it is homozygous, it is like this. Okay, Antonia B, don't get confused. Yes, solve this for me. So solve this for me. Yes, this is how you solve it. I hope everyone understood how to find the gametes. Yes, is this clear? So do it slowly but gradually improve your speed. So initially everyone cannot do it fast, it is tough. But gradually, yes, everyone can increase your speed. Yes, now tell me the genotypic and the phenotypic ratio. So this cross is clear? Yes? Fine. So I will write down which is what character. So this is round yellow, this is yellow round, this is also yellow round, this is also yellow round, yellow and round. right, looking at the genotype. So this is also yellow round, this is yellow but it's wrinkled, so this is yellow round, this is yellow but it's wrinkled, right? Yes, so this is yellow round, so this is green and round, so this is also green and round, right? Okay, so this is your yellow round, yellow wrinkled, so green and round, so this is green and wrinkled. So you have only two. How many phenotypes you have? We have 4 different types of phenotypes. So I am writing the phenotypic ratio for yellow round, that is the dominant character. 1, 2, 3, 4, 5, 6, 7, 8, 9. So this is 9. Okay. So now I am writing for your yellow wrinkled. So how many are yellow and wrinkled? So 1, 2, 3. So this is 3. Now the next one I am writing is green and round. So green and round is 1, 2 and 3. And the last one is green wrinkle, so that is 1. So my phenotypic ratio is 9:3:1 for my dihybrid cross. So did everyone understand how did you get 9:3:1? So whatever I have written in green, that is my genotype. The physically how will the plant be? Whether it will be yellow color or round color, green color or wrinkle color. Is this clear? Yes to everyone? Alright. So now talking about your genotypic ratio. So what is genotypic ratio? So this is a bit complicated because you have a lot of varieties here. Okay. So capital Y, capital Y, capital R, capital R. So this is repeated only once. This is homozygous dominant and this is homozygous recessive, right? So I write 1. So homozygous dominant, homozygous recessive. So this is done. Clear? Okay. So now capital Y, capital Y, capital R, small r. So this is repeated twice. Yes. So I write 2 times. Okay. Yes, but at some point your triangle Punnett square is pretty tough. Yes, if you are clear with that, you can do, but let me teach it systematically. So then if you find shortcuts, then that shows your confidence. So then this is same, this is same, right? So it is again 2. This is the odd one out, so this is 1, right? So again this is same, this is same. So these 2 I am cancelling, so 1:2. So then this is the different, so it is 1:1. Okay, so this one I have written, so this and this are the same, this and this are the same. Okay, so 1:2, then again this is different. So this will be my genotypic ratio. Okay, so you can actually see it here. Yes, so this is the cross. Okay, so here they have discolored it, so it will be kind of easy. So 9:3:1 is my genotypic ratio, 1:2:1:2:4:2:1 is my genotypic ratio. So if you are still confused in this, I can tell you again. Is there anyone who is still confused? Yes, till this it's clear to everyone? Yes, no doubt? Yes, questions we are going. So for all those who are listening, so this is 1, 2, 1, 2, yes. So till this it's sorted and this is 1. So again, so this is repeated 1, 2, 3, 4. So this is repeated 4 times. So 1, 2, 1, 2, 4. Okay, so this is my genotypic ratio. Okay, fine. So with this your dihybrid cross is done. So now based on this you have the last law of Mendel. So when two pairs of traits are combined to form a hybrid, yes, so we have seed color and seed shape, right? So when they are combined in a hybrid, segregation of one pair of character is independent of the other pair of character. Okay, for example, it can be round or yellow, or it can be green or round. So this segregation of two characters is independent of each other. So this is your law of independent assortment. Okay, so I have taught you in a systematic way. So now I hope you can solve questions. Before that, Anthony Abhi wanted me to explain what is mutant allele and what is wild type. So wild type is what is present in majority of the population. So majority of the Indians are tall. So wild type population is tallness. So mutant allele is one which is present in less than 1% of the population. Okay. So less than 1% of the population. So that is your mutant allele. Okay, for example, less than 1% in the population you have recessive genes that is expressed. So this is wild type and this is mutant allele. Alright, yes? Okay? Okay, so I hope everyone is clear. Yes, I know I have to do questions and questions are here. So when you are in a class having so many different people, there are people who are watching it for the first time. There are people who are 11th standard aspirants. There are people who are target students as well. But yes, I can go according to satisfy everyone's requirements. I cannot just skip because you know it. Right? So here a lot of people are listening to me, not only you. So you have to be patient. It's not your personalized class, right? So please hold on, have some patience. I cannot rush. There are even 11th standard students who are listening to it. Yes, I know you know it. Yes, even I know. Then why should I teach? But I cannot rush, right? So please hold on, have some patience. Yes, I am going to solve questions. I'm not going to run away. But if I'm explaining something, just because you understood it, don't be in a hurry. So always have patience. So patience will take you to a better place. Okay. So it is not just me, Vishal, Keshav who's in class. So I assume, okay, so Vishal and Keshav are good students. They know. But here I have a lot of them, right? So this is the open channel. Yes, the questions are here. Come on, quickly give me the answers and please evaluate yourself at the end. Tell me your marks as well. The recombinant phenotypic ratio in F2 obtained from parental cross having genotypic ratio will be. So I want only the recombinant phenotypic ratio. Not the entire phenotypic ratio, but only the recombinant phenotypic ratio. Yes, make it quick. Come on. Yes, because that is psychologically been found. So before every class, if you recap the previous class for 15 minutes, students tend to remember the concepts much quicker. They tend to retain the concepts much quicker. So that is why I follow. So in a lot of very good institutes like your IITs, so they do this. Option A, I want only the recombinant phenotype. Satinder, that's why I said read the question. I want only the recombinant phenotype. So I am solving this. So they are saying F2 generation. So I have to solve it. So this is my parental. So these are my gametes. So my F1 will be right. So this is normal monohybrid cross. So phenotypic ratio is 9:3:3:1. So 9 is what? Yellow round. Okay, according to our dihybrid cross, right? So here it will be tall and round, right? So here it will be dwarf and recessive. These two are the recombinant ones, right? So this will be tall and small t. So this will be dwarf and capital R, right? Yes, so the recombinant is 3:3. I want only the recombinant, the new varieties. So it is option D. Yes, Keshav, you are absolutely right. So others are able to understand why it is D. So Shimpi, it is D. So read the question. This is how you go wrong. Okay, done? So shall I move forward? So question number 2. So total 512 seeds are collected from the cross this. Okay, so find the number of plants produced with the first dominant and the second recessive trait. Let's see how many of you give me the first answer, correct answer, then I will move forward with it. Yes, then I will move forward and I will go forward with it. Yes, let's see who gives me the right answer. So I guess it should be capital Y, small y. Yes, so this is the cross. Yes, Shimpi, perfect, very good. How come you gave me the right answer in one go? Very good. How did you solve? Yes, Vishal, very good. Okay, when you see this type, your mind goes blank. Okay, relax. I will tell you how to solve. So, from this cross, so it is a dihybrid cross, it is understood, right? So, they want first dominant and second recessive trait. So, what is the first dominant? So, this is the dominant one, right? And the recessive one is Y. So, you have to count all of these types, okay? So, all of these types, how many are there? So, this is what? So, this is dominant, recessive. So, you have 3 plants which comes under this category. So, 9:3:1. So, this is homozygous dominant, this is homozygous recessive, and one is in this condition, the other one is in this condition. Right. So, this 3 is of this condition. So, totally we have 3 plants like this. So, 3/16 offsprings, okay, into 512. So, this gives me 96 plants for all those who haven't understood. So, this is how you solve this question. Okay, fine. What is this calculation? So, this is this calculation. Okay, Keshav, is it clear? Is it clear now? How 96? Yes, any more doubts? Keshav, you understood? Okay, so moving forward to question 3. So, from this cross genotypes, what is the ratio of these genotypes? So, what is the ratio of these genotypes? Yes, quick. What is the ratio of these genotypes? Come on, let's see who gives me the first correct answer. Yes, quick. Whom are you waiting for? Make it quick. Okay. So, total in your dihybrid cross, you have 16 offsprings, right? 9 plus 3 is 12, 12 plus 3 is what? 15, 15 plus 1 is 16. So, 3 are of the type that we need, the total is 16 and the number of seeds that they have given is 512, so it is 96. Alright Keshav, did you understand why it is 3/16? So, 3 plants here are according to our need, first dominant and second recessive. The first character is dominant, the second character is recessive. Okay Keshav, did you understand? Yes, can I go to the next question? Yes, others give me the right answer for this question. Quick, make it quick. Yes, make it quick. Alright Keshav, you understood. Yes, what is the answer for this question? Come on, quick. Yes, you're right. Shimpi says A. No, A is the wrong answer. Yes, others? A is the wrong answer. Solve this. So, what will be the gametes here? Capital A, capital B, capital A, small b, right? Here. Yes, Antonio B. B is the right answer. Very good. So here it will be small a, capital B, small b, small a, small b. So now do the crossing. AB, AB. Right. So this is the answer. So they have asked this. So this, the ratio for this is 1 is to capital A, small a, capital B, small b. So this is 2. So A, B, B. So this is 1. There is none of this type, right? So it is 0. So it is option B. Yes, is it not a easy one? Your messages are deleting. I cannot see any of your messages, Shimpi. You said A option? Yes, only that I saw. Okay, so I am doing this cross. I found the gametes for this parent. So capital A into small b. So capital A is homozygous. So only one A will be my gamete. So capital A into capital B, capital A into small b. Right? So I have two gametes. Then small a into B, small a into B. So two gametes. So I do the cross. Yes. So according to the given question, I match the ratios. So this is 1, this is 2 and this is 1 and there is nothing of this genotype. So it is 0. So option B is the answer. So Vishal, did you get it? Yes? No doubts? Is this clear? Did you get it? How it is option B? No doubts? Keshav, Vishal, Shimpi. Nightboat Bhaiya is deleting your message. Okay, so did you not listen to Nightboat? So Nightboat is angry with you. But here it does not show it is messages are deleted. Easy, but more practice needed in this area. I don't touch this area yet. Okay, so at the end of this PPT also you have homework questions, so please solve and come. So tomorrow we will discuss in class. Okay, so in a cross between pure tall pea plant with green pod and a pure plant short plant with yellow pod, how many short plants out of 16 would you expect in your F2 generation? Yes, quick. How many short plants would you expect in the F2 generation? So the answer for question 4, yes, quick. Yes, quick. Come on, please do like, share and subscribe the session. Yes, what is the answer? So tall plant with green pod. So it is 2 characters again. So Shimpi says C is the wrong answer. Yes, Vishal, you are absolutely right. B is the right answer. So it is dihybrid because you have 2 characters, tall and green pod, right? So tall, green pod, so dwarf and yellow pod. Okay, so these are the parents. So the gametes will be TG, TG. So F1 progeny will be like this. So when I do selfing, I will get 9:3:1. Okay, so this will be tall and green pod. So this will be dwarf and yellow pod. Okay, so this will be tall with yellow pod. So this will be dwarf with green pod. So the tall plants are 3 plus 1, that is 4. So your option is B. Why? Even these 2 are tall. So it is 4. Option B, 4 is the right answer. So did you understand? Yes, in 2 small r's and 2 small y's, it is only recessive. Yes. No, I have 2 tall plants. They are only asking about 1 character here. They are asking how many short plants I will have. I will have 2 short plants irrespective of the pod color. Yes, clear? Did you understand, Keshav? Okay. So you people find it tough still. Yes, question 5, quick. So in mice, black coat color is dominant over brown coat color. The offspring of a cross between, they are saying the offspring of a cross between black and brown were allowed to interbreed. So what percentage of the progeny would have black coats? Yes, quickly solve this. Come on, quick. Yes, start. Yes, what is the answer? Question 5. Come on. What will be the answer here? Yes. Should I solve? Yes, one of you give me the right answer. Yes, Satyendra says. Okay, so Shimpi says C, Satyendra says C, yes, absolutely right. So it is 75%, that is the right answer, very, very good. Yes, Anthony Abhi has also given me the right answer, good. So which means it's clear? Okay, so once again for all those who could not solve, so you have black coat and brown coat. This is monohybrid cross, okay, so they are the parents. So here the gametes will be B and small b, right? So this, okay, so the gametes will be B and B. So the offspring F1 progeny will be capital B small b. So the offspring of a cross between black and brown. So black and brown, so this is the offspring. Now they are allowed to interbreed. So if they interbreed, it will be like this. This is your monohybrid cross. So what is the percentage of progeny with black coats? So this is black, these two are black. So you have 4 which makes 100%. So this is 25, 25, 25, 25. So 3 of them are black coats. So I take it as 75%. Okay, yes? Yes, yes. If they ask only with yellow, then it is 1. You started eating noodles. Oh my God. Okay, so you are very hungry, alright. Yes, as long as you give me the right answer, I don't mind. So everyone understood how it is 75%? Okay. So question 6. So in a Mendelian dihybrid cross, when heterozygous round yellow are self-crossed, ground green offsprings are represented by the genotype. So it is directly given Mendelian dihybrid cross. Yes, what is the answer for question number 6? What is the answer for question number 6? Yes, quick. Yes, even I will be hungry. Yes, Keshav, you are very true. Yes, now even I have the mind for eating noodles. But recently there was an article, right? So 75% of all the Nestle items, including Nestle coffee, Nestle chocolates, biscuits, everything is not healthy. So that was a big shock because I have a lot of Maggi packets. Yes, option A. Seriously? This is the wrong answer. D. D is also the wrong answer. Why confusion? In Mendelian dihybrid cross, so in a dihybrid cross, so you have heterozygous. So dihybrid cross has this, right? So if they say heterozygous, it is like this. So now this is self-crossed. So this is selfed in your F1 progeny. So the round green offsprings are represented by the genotype. So look at the offsprings. So they want round and green. So this is round, this is green. So capital R is for round and this is for yellow. This is for green and this is for wrinkled, right? So this is, you want round green, right? So round green, round green. Wait, so this is round yellow, this is round yellow, so this is also round yellow. So round green, but this is not round, so this is wrinkled green. So this is also not the answer. So this is round green, okay. So this is wrinkled green, this is wrinkled green. Yes, this is the answer, option D is the right answer. So this is your round green, yes, Absolutely. I am sorry. I thought option B is the answer. Yes, option D is the right answer for question number 6. Yes. Yes, yes. So D is the right answer. Okay. Yes. So question 7. Here we move forward to question 7. How many types of gametes can be produced by a diploid organism whose heterozygous for 4 loci? Yes. You were confident. All right. Very good. Yes. So diploid organism for 4 loci. Come on, quick. Diploid organism 4 loci. So 4 into 2. Sorry. 4 square 16. So option C is the answer. 4 loci means 4 locations. Okay. So if I have a gene like this, so it will be what? So what is 2 power 4? This way also you can do. This way also you can do. It is option C, that is 16. Okay. Yes. So I have 15 questions. So you have 8 more questions as homework. So 4 loci means 4 different locations. I am having 4 different genes. Okay. So how do you find the gametes? 2 power 4. So if it is like this, it is 2 power 1. So I have 2 gametes. So if it is like this, so I have 2 power 2. So I have 4 gametes. So similarly, it's 2 power 4. So that is 16 gametes. Alright. Yes. So how many types of gametes can be produced by a diploid organism whose heterozygous? So they are heterozygous for 4 loci. In 4 different loci, I am having 4 different genes. Okay. Yes. 2 power n. Okay, so there I will discuss 30 questions. So please be there for the next session at 6 p.m. So one more thing, I have totally 15 questions. I have solved only 7 questions. So the remaining 8 questions will be your homework. So please download my PPT. So solve the remaining 8 questions and come for the next class. So your next class will be on Wednesday for genetics. So we are meeting again at 6 p.m. So we are discussing 30 important questions from Cell the Unit of Life. So I will go pretty fast. Okay. Yes. So please join me for the same, and thank you so much for joining my lecture. I hope you understood. If you genuinely liked my way of teaching, so please do like, share and subscribe. So this is me wishing you more and more. So take care and bye.